So multi-cancer early detection is an exciting new way of screening men and women for cancers. And let me be clear, it's not specifically about prostate cancer. It's about the broad screening paradigm. In the United States, we currently screen for 5 cancers: breast, colon, lung in high-risk patients, cervical, and prostate cancer. And we do that because we know that screening reduces mortality, and that's a good thing. The problem with that paradigm, though, is that even though we do all of that, more than 600,000 people a year die of cancer in the United States. So there's real opportunity to do better. So we have this new technology, which is based on next-generation sequencing, the same technology that was used to sequence the human genome. And we use something called liquid biopsy. It's possible to take a blood sample and detect what's called circulating cell-free DNA, which is shed by our cells. And in the background of all the normal cell-free DNA that's in our system, we can find a cancer signal in individuals who have cancer. And that's how sophisticated the technology is. So you can measure a particular target in cell-free DNA, methylation or fragmentomics or mutations and so forth. And you can sort out, again, from all that background noise, whether or not a cancer signal is present. So in some of the studies that I presented from a company called Grail, we were able to identify a cancer signal in about half the patients who had cancer across all stages. We were able to identify 80% of ovarian and pancreatic cancers for which there's no current screening paradigm. We were able to detect about 60% of stage 1 and 2 pancreatic and ovarian cancers, but we were really able to detect more than 50 cancers. So this test and others like it by other companies are being developed to be used ultimately in the general screening population. To get back to the initial observation that I made, one main reason that 600,000 people a year still die of cancer, even though we screen for 5 of them, is that those 5 cancers don't kill the majority of people who die from cancer. And so these blood-based next-generation sequencing tests are a way to broaden our ability to detect cancers that are actually clinically significant. So I'm pretty excited about all of that.